Now, in the previous meeting, we have learned of the glycolysis as well as the citric acid cycle, and we've learned that these pathways synthesize or lead to the formation or to the production of reduced equivalents, nicotinamide adenine dinucleotide, as well as, of course, ATP. Today, we're going to consider exactly the fate or what happens to nicotinamide adenine dinucleotide, and in the process, be able to figure out how much ATP is indeed formed during aerobic metabolism or aerobic respiration. Okay. Now, the discussion will primarily be focused on the activity of the electron transport chain as well as that of the synthesis of ATP through oxidative phosphorylation. Alright, now the electron transport is carried out by, as you can see here, by 4 closely related multi-subunit membrane-bound complexes and 2 electron carriers, coenzyme Q and cytochrome C. Now, the multi-subunit membrane-bound complexes are actually consist of a number of different proteins. This can be heme, iron heme containing proteins, and or iron sulfur clusters. The important thing to remember is the fact that this membrane-bound complexes have the ability to accept electrons and to transfer electrons among the component proteins, specifically the iron in the heme or the iron in the iron sulfur complexes, and ultimately to coenzyme Q and to cytochrome C, and ultimately to oxygen for that matter. Now, coenzyme Q, these are mobile electron acceptors and electron donors. They are electron carriers, but unlike the membrane-bound complexes, are capable of moving through the inner mitochondrial membrane. The same is true with cytochrome C. The membrane-bound complexes here, the multi-subunit membrane-bound complexes, on the contrary, are relatively, I'm not going to say that they're immovable or not moving, because you've already known from the fluid mosaic model that the proteins have this ability, and the phospholipids have this ability to move laterally in the cell membrane. I'm just going to say is that the coenzyme Q and the cytochrome C are definitely more mobile, in the sense that they are they are smaller. And also another thing to bear in mind is the fact that coenzyme Q and cytochrome C, this do not consist of multi-subunit proteins. Coenzyme Q, for example, is ubiquinone. It's a small molecule. Okay. In a series of oxidation-reduction reactions, the electrons from the flavine adenine dinucleotide and nicotinamide adenine dinucleotide, these are the reducing equivalents, or the reduced equivalents, but one obtained from glycolysis as well as from citric acid cycle, are transferred from one complex to the next until they reach oxygen, the ultimate and the consummate electron acceptor. The oxygen is reduced to water, so respiration produces water, as you might already be aware of. As a result of electron transport, the protons are pumped across the inner membrane to the intermembrane space, creating a pH gradient. This is the link between the electron transfer chain as well as the synthesis of ATP through oxidative phosphorylation. The generation of the proton gradient, or the pH gradient, an electromotive gradient across the inner mitochondrial membrane, which ultimately allows for the synthesis of ATP. Okay, we're going to discuss the details of each of these, so do not worry. Okay. The production of ATP in the mitochondrion is a result of oxidative phosphorylation, and I must say the yield of ATP in the mitochondrion is a result of oxidative phosphorylation in electron transport chain is vastly greater is so much greater than the yield of ATP from substrate level phosphorylation, for example, in glycolysis, in anaerobic glycolysis. The proton, so the production of ATP in the mitochondria is a result of oxidative phosphorylation, and the proton gradient, as I've already mentioned, establishes a voltage gradient across the inner mitochondrial membrane. And this voltage gradient ultimately allows for the return of the protons, which is coupled to the synthesis of ATP. We're going to learn more about that later. The proton and the voltage gradients together provide the mechanism to couple electron transfer with phosphorylation of ATP and the concomitant synthesis of adenosine triphosphate, or ATP. Okay, here is the go to some basics about the establishment of a proton gradient. Here is the mitochondrion. All right, so we have an outer membrane, outer mitochondrial membrane, and an inner mitochondrial membrane. The electron transport leads to proton pumping across the inner mitochondrial membrane. So, inner mitochondrial membrane, the protons are pumped out of the matrix, the mitochondrial matrix, into the intermembrane space, creating a region where you have a high concentration of protons or hydrogen ions in the intermembrane space, in other words, low pH, and low concentration of hydrogen ions inside the mitochondrial membrane, in other words, in the matrix, which have a high pH as a result. Okay, the mitochondrion takes advantage of this proton gradient in the synthesis of ATP for oxidative phosphorylation. I'm going to learn the details of that later, so do not worry. The electron transport from one carrier to another, like, for example, we're going to discuss the details of this, so do not worry. So the electron, this is just an overview of what we're about to discuss in detail. So, for example, the reduced equivalents like NADH as well as FADH2, these are oxidized through a series of electron loss and electron gain, in other words, a series of redox, reduction, oxidation reactions, but involve a number of different intermediates like flavine mononucleotide, coenzyme Q, cytochrome B, cytochrome C1, cytochrome C, cytochrome AA3, till ultimately the electron is transferred to oxygen. There are 4 complexes involved in this case, but of the 4, only 3 involve proton pumping. And of these 3, they transport protons from the matrix to the intermembrane space. So the transfer of electrons from one carrier to another, say for example from NADH to FADH2 through this intermediates and ultimately to oxygen, leads to the creation of a proton gradient through pumping of protons from the matrix into the intermembrane space. As we've said here, the electron transferred from one carrier to another creates a proton gradient across the inner mitochondrial membrane. Now, the proton gradient is coupled to the production of ATP in aerobic metabolism, right? Okay. Now, useful way to look at electron transport is to consider the change in free energy associated with the movement of electrons from one carrier to another. Right? If we have 2 electron carriers, for example, NADH and coenzyme Q, just for example, are electrons more likely to be transferred from NADH to coenzyme Q or vice versa? And in this case, our knowledge of reduction potential will be very useful. What we need to know is the reduction potential for each carrier. Remember that reduction potential is a measure of how easily a substance accepts electron. In other words, how good it is at accepting electrons from another substance, and therefore it's a measure of its ability to act as an oxidizing agent. Yes, it is reduced in the process, but also in the process it causes the loss of electron of another substance, so it oxidizes another substance. It is the oxidizing agent, in other words. So a carrier of high reduction potential, in other words, that which is very good at accepting electrons, it has high reduction potential. When you say high reduction potential, it means that we have a positive reduction potential, will tend to be reduced if it is paired with a carrier of low reduction potential. So, for example, you have here question ethanol to acetaldehyde. The reduction potential here is the question is, how do you measure reduction potential? what we do typically is to measure or to create a cell, an electrochemical cell, in which the reference is hydrogen, solution containing hydrogen ions at 1 molar concentration, and which is in equilibrium with hydrogen gas at 1 atmosphere pressure. And then you connect that with a solution containing, for example, the redox couple, or rather the substance that you want to either undergo oxidation or reduction. So in this case, we're measuring the reduction potential of acetaldehyde, and we found that when you couple a mixture of acetaldehyde and ethanol, both acetaldehyde and ethanol, by the way, are at 1 molar concentrations, we found that what happens is what happens is that if you're going to look at this, look at the value. It's negative, right? It's negative 0.197 volts. Now, what does this mean? This means that the reduction potential of acetaldehyde to ethanol is lower than the reduction potential of hydrogen ion to hydrogen gas, which is given a value of 0. Now, this effectively means that the ethanol is preferentially oxidized into acetaldehyde, and not the other way around, not acetaldehyde being reduced into ethanol when it is coupled with the reference hydrogen ion hydrogen gas electrode, which has a 0 reduction potential. In this case, then the hydrogen ion hydrogen gas acts as the cathode, right? It acts as a cathode where reduction occurs, and the ethanol acetaldehyde electrode, by the way, the cathode is an electrode where reduction occurs, while in this case the one in which oxidation occurs is called the anode. So in this case it's ethanol acetaldehyde pair that acts as the anode. Okay, and it has, notice that acetaldehyde to ethanol has a negative reduction potential. That again means that ethanol is preferentially oxidized to acetaldehyde when it is paired with a hydrogen ion hydrogen gas electrode. Now, a different case is observed in the case of fumarate to succinate. When fumarate to succinate, or fumarate and succinate, half cell is paired with the hydrogen ion hydrogen gas half cell, the fumarate to succinate has a positive reduction potential, which means that fumarate is preferentially reduced into succinate when it is paired with hydrogen ion hydrogen gas half cell. Now this means that fumarate is a better oxidizing agent than hydrogen ion, and so fumarate will oxidize hydrogen gas to hydrogen ion. Okay, so as a rule, if you want to know which of the substances is going to be reduced and which of the substances is going to be oxidized, you just need to look at the values of their reduction potential. For example, let us look at the first two instances. 1/2 oxygen, 2H plus 2 electrons forming water. The reduction potential is 0.816 volts. The reduction of iron 3 to iron 2 plus has a potential of 0.771 volts. So that means that the oxidized form of iron, Fe2 plus, has a lower reduction potential than the oxidized form, than the oxidizing agent in the first reaction, which is oxygen. So this means that oxygen will have the higher reduction potential and will therefore oxidize the reduced form of iron, which is Fe2 plus. Remember, the one which has a higher reduction potential will preferentially act as the oxidizing agent. Alright, so if you're going to look at this, it is oxygen that is the ultimate or the consummate oxidizing agent because it's the one with the highest reduction potential. Now, if we're going to look at NAD plus and NADH. Remember, NAD plus is the oxidized form of NAD, and NADH is the reduced form of NAD. And therefore, it is NAD plus that's going to act as an oxidizing agent, and NADH is going to act as a reducing agent. The same is true with FAD and FADH2. FAD is the oxidized form of FAD, and FADH2 is the reduced form of FAD. And therefore, FAD is going to act as the oxidizing agent, and FADH2 is going to act as the reducing agent. Alright, let us look at one example in this case. If we're going to look at this, let's look at the case of oxaloacetate being reduced to malate, as well as the NAD plus being reduced into NADH. Which of the two has a higher reduction potential? For oxaloacetate being reduced to malate, its reduction potential is negative 0.166. The reduction potential of NAD plus being reduced to NADH is negative 0.320. Remember, so notice that the oxaloacetate has a higher or the more positive reduction potential and will therefore be preferentially reduced by NADH. Alright? Remember, the more positive it is, the better it is as an oxidizing agent. And remember that the flow of electrons is from substance with a lower reduction potential to the substance with a higher reduction potential. That will be the logic in our study of the electron transport chain. Standard reduction potentials provide the basis for a comparison among redox reactions, and that the sequence of reactions in the electron transport chain can be predicted by using the reduction potentials. The one with a higher reduction potential is the one that's going to act as an oxidizing agent. Or ultimately, you can say that the electrons will ultimately be transferred from those with lower reduction potentials to those with higher reduction potential. And that is why oxygen is the ultimate electron acceptor in this reaction in aerobic respiration, because it has the highest reduction potential, the most positive reduction potential of 0.816 volts. Okay, now as I've mentioned, there are a number of different complexes in the electron transport chain. These are the proteins or the multi-subunit protein complexes. Not just multi-subunit protein complexes. They consist of a number of different proteins which mediate the transfer of electron. Okay, so in the first complex, complex 1, we call that NADH coenzyme Q oxidoreductase. The electrons are passed from NADH to FMN, to flavine mononucleotide, right? And in the process, the flavine mononucleotide is reduced to FMNH2. This is the flavine mononucleotide. It does not have the adenine, so that's why it's called flavine mononucleotide, right? Now, the electrons from FMNH2 are then transferred past the iron-sulfur clusters, and the last step of the complex 1 involves electrons being passed to coenzyme Q, also called ubiquinone. So this is the complex 1. You can think of this as this is the electron transport chain, all right? So NADH transfers its electron to FMN, and then FMNH2 transfers its electron to the iron-sulfur cluster. When you have Fe-S, that's the iron-sulfur cluster proteins, and you have OX there, which means it's the oxidized form, so it is reduced as a result. The reduced iron-sulfur cluster then transfers its electron to ubiquinone or to coenzyme Q, process producing reduced ubiquinone. So the complex 1 is that which oxidizes NADH and which ultimately reduces coenzyme Q. Okay. Now, I'm not sure. Okay, we're going to discuss complex 2 later. So there are a number of different complexes, as you can see in this case. This is complex 1, complex 2, complex 3, and then complex 4. Alright, the transfer of electrons is strongly exergonic and is sufficient to drive the phosphorylation of ADP. For example, the NADH being oxidized and ultimately transferring its electron to coenzyme Q releases negative 81 kilojoules per mole of energy, while cytochrome B transferring it to cytochrome C and ultimately to cytochrome A, negative 34.7 kilojoules per mole. And that from cytochrome A to oxygen, it releases negative 102 kilojoules per mole. If I'm not mistaken, the amount of energy that is necessary to generate ATP or to synthesize ATP is just around 30 or so kilojoules per mole. So this amount of energy are definitely large enough to synthesize ATP, right? Okay. We're going to learn exactly how ATP is synthesized later. So the complex 2, I'm not sure if you've seen that here. There's a complex 2. The complex 2 is actually the succinate coenzyme Q oxidoreductase. And this complex 2 actually has as one of its components the enzyme that is part of the citric acid cycle. Remember that which oxidizes succinate to fumarate? It is here. So succinate is oxidized to fumarate. In the process, FAD is reduced to FADH2, right? And then FADH2 is then again oxidized with the iron sulfur clusters in the process producing coenzyme Q as a result. Okay, so notice that here you generate coenzyme Q as well. Here you also in complex 1 you also generate coenzyme Q as a result. So you can think of complex 1 and 2 as both increasing the pools of coenzyme Q or ubiquinone that is ultimately oxidized in complex 3. All right. All right. Okay. So again, you start here NADH. The NADH here is the one that comes from the citric acid cycle. And FADH2 here is the one which comes from the citric acid cycle also. But remember, that's the enzyme, remember that the succinate dehydrogenase enzyme, the enzyme that oxidizes succinate to fumarate, is actually part of the complex 2, the succinate coenzyme Q oxidoreductase complex, which is itself found embedded in the inner mitochondrial membrane. Okay, we're going to have more to say about the ATP yield, the P to O ratio later. Okay, now complex 3, as you can see in this case, ultimately oxidizes the coenzyme, the reduced ubiquinone, which is the reduced form of ubiquinone, coenzyme Q, to iron sulfur cluster, ultimately reducing cytochrome C1 into cytochrome C1 reduce. And this cytochrome C1 reduce will then reduce cytochrome C, the oxidized form of cytochrome C, into cytochrome C. And this cytochrome C, as I've mentioned, is mobile. It will ultimately, it moves around the inner mitochondrial membrane. Okay. Complex 3 involves ultimately the oxidation of coenzyme Q, reduced coenzyme Q or ubiquinol, or reduced ubiquinone into oxidized ubiquinone, forming cytochrome C, which is reduced. Now, cytochromes are heme-containing proteins, and the heme, this similar to the heme found in hemoglobin. The only difference is that the iron of the heme in this case do not vacillate. In the case of the electron transport chain, do vacillate. They change from Fe2 to Fe3 because they act as electron acceptors. In the case of the heme, in the hemoglobin, such change is not happening. It's always in one oxidation state. All right. Okay. Now, the heme will have, as I've mentioned here, because you're going to see different kinds of cytochromes: cytochrome C1, cytochrome C, cytochrome A. These different cytochromes only differ in the structure of the hemes, and also differ in some subtle differences in the structure of the proteins in which they are found. And this differences is responsible for the differences in their reduction potentials. Remember that ultimately, the electron is transferred from the one with the lower reduction potential to the one with higher reduction potential. So that change in the reduction potential is, of course, attributable to the changes or differences in the structure of the heme in which the iron is found, as well as the protein in which both the heme and the iron is found. Okay. So in complex 3 coenzyme Q, the reduced ubiquinol or ubiquinol is oxidized back to ubiquinol in the process, reducing cytochrome, which you have cytochrome C, in which iron is in the +3 oxidation state to +2 oxidation state. The reaction to this complex results in decrease in free energy that is sufficient to drive the phosphorylation of ADP to ATP. All right. The flow of electrons from reduced coenzyme Q, a quinone that can exist in free form, is known as the Q cycle. Now, here, this is the flow of the electrons that I want you to notice. It's the compositions and the locations of the respiratory complexes in the inner mitochondrial membrane. Again, the NADH is oxidized by complex 1, passing, remember, 2 electrons are transferred to the flavine mononucleotide, transferred to the iron sulfur clusters, which is transferred to the molecule of the ubiquinone coenzyme Q, and then this coenzyme Q is oxidized by complex 3. This complex 3 is oxidized how? Again, iron sulfur, cytochrome. This is a complex process. There are a number of different steps in this case, passing through electrons pass through iron sulfur cluster, ultimately to cytochrome C1, and then ultimately to cytochrome C. Okay, but there is also a Q cycle where the coenzyme Q is oxidized by cytochrome Bl and then transfers its electron. The reduced cytochrome Bl reduces oxidized cytochrome BH, and then cytochrome BH reduces the coenzyme Q. And this is called the Q cycle. That Q cycle is shown, the phenomenon, or rather the preponderance of the Q cycle is attributable to the fact that the coenzyme Q exists in the form of ubiquinol, as well as the semiquinone, and the semiquinone is involved in the Q cycle because that one involves the transfer of 1 electron at a time. Okay, now the cytochrome C that is produced in complex 3 travels through the inner mitochondrial membrane until it encounters complex 4. In complex 4, notice that its cytochrome C is oxidized by cytochrome A, which is oxidized by cytochrome A3, which ultimately causes the reduction of oxygen into water. All right. Okay. Now the complex 4 is called cytochrome C oxidase, and it catalyzes the final step in the electron transport, where you have cytochrome C in the form of with its iron in the reduced form Fe2 is ultimately oxidized by oxygen to in the process produce oxidized cytochrome C, have iron 3, and water as a result. This is the overall reaction, again because the reaction actually happens through a number of steps where cytochrome A is reduced, cytochrome A3 is reduced, and then ultimately oxygen is reduced to water. Alright? Okay, complex 4 contains cytochrome A, cytochrome A3, and copper 2, which are also involved in the electron transport. So complex 4 is a link to molecular oxygen. Alright? So here are the energetics of the transport reactions. And you can see here the oxidation of NADH to NAD+, the reduction of FMNH, of FMN to FMNH2, and so on and so forth. The overall reaction, ultimately that of the oxidation of NADH by oxygen, is exergonic. You have negative 220 kilojoules per mole of energy, or per mole rather, of NADH is formed. Right? Now, as I've mentioned a while ago, all of the cytochromes contain a heme group, and there are differences in the side chain depending on the heme. Alright? Okay. Okay. Now I'm not sure. Oh, it's not shown here. I'm trying to see. Alright. It will be shown later. Okay, it will be shown later. We have been asking ourselves a question like, how then is the electron transfer chain related to oxidative phosphorylation, that is, to the synthesis of ATP for the process of oxidative phosphorylation? The answer is that along the way, when the electrons are transferred from NADH to ultimately oxygen, along the way in these complexes, the protons are pumped from the matrix to the intermembrane space, creating a gradient. And it is this gradient that develops potential energy, which is ultimately harnessed in the form of mechanical energy, and then ultimately in the form of chemical energy in the form of ATP. Okay, that is the thing that we're going to study later in detail. But for now, the connection between the electron transport and phosphorylation is as follows. The energy releasing oxidations give rise to proton pumping. Again, when we say proton pumping, it's the somewhere along this or in this complex as the protons are transported from the inner membrane to the outer membrane. It's like you're building a dam. I mean, it's like you have a dam, right? And you're filling the water up to the height of the dam, and that develops the height of water develops energy. There's a lot of potential energy associated with that. And so when you release the water in a dam, that, excuse me, is capable of generating electricity, right? Pushing a rotor, I mean, pushing a rotor, and then um, the energy is ultimately converted to electricity. The energy-releasing oxidations give rise to proton pumping in a pH gradient across the inner mitochondrial membrane. The differences in the concentration of ions across the membrane generates a voltage gradient. So the potential energy here is not just due to the differences in the concentration of the hydrogen ion, but also to the fact that you end up with the differences in the charges, because the protons have charges, or hydrogen ions have charges. The coupling process converts the electrochemical potential to the chemical energy of ATP, and the coupling factor, the one which couples this or which accomplishes this, is the ATP synthase, a complex protein oligomer separate from the electron transport complexes. What it effectively does is it harnesses the energy of the electrochemical proton gradient and converts that in the form of chemical energy in the form of ATP. Uncouplers inhibit the phosphorylation of ADP without affecting the electron transport. The examples are, of course, 2,4-dinitrophenol, valinomycin, and gramicidine A. They uncouple the electron transport from the synthesis of ATP. We're going to have to learn more about that later. This is the ATP synthase, and we're going to go back to this later. Notice that the ATP synthase consists of this. This is a protein. So this one is F sub 0, the one which is embedded in the inner membrane, while this is F1. This F1 is actually found in the side of the inner membrane space, but is exposed to the matrix. All right. So you can think of what's happening this way: the protons, which have high concentrations in the intermembrane space, enter the F sub 0, in other words, this one, F sub 0, and what happens? They bind, or rather move, they move through the F sub 0 protein, like, can you follow that? They move through the F sub 0 protein until ultimately they are, I'm sorry, because this is the outer membrane, the intermembrane space. So the proton enters through here. You have a high concentration in the intermembrane space, and then they enter through that, and then they move through that channel, ultimately being expelled into the interior or into the matrix. And their movement causes, or the translocation of the protons from the intermembrane space to the matrix, ultimately causes the rotation of this rotator like epsilon gamma protein, ultimately causes the movement, the rotation of that protein, which then causes the alpha beta subunits to change in conformation. And these alpha beta subunits are actually the one responsible for the synthesis of ATP. We're going to learn more about that later, but for now, the important thing that you have to bear in mind is the fact that the concentration of the hydrogen ions outside are high, and so the protons tend to come back through the this hole in the F sub 0, the ATP synthase, and their movement will ultimately cause the rotation, like, because they are moving there, like they are being funneled back into the matrix, and their movement of a translocation of protons will ultimately lead to the rotation of this rotator, which then will affect the structure of the alpha beta subunits, which will then cause them to change from the open to the tight to the loose binding conformations, which are responsible then for the synthesis of ATP, the details of which will be discussed later. Okay, Here, the 2,4-dinitrophenol, this is the valinomycin as well as gramicetine. 2,4-dinitrophenol actually exists in the form of its conjugate base, like it's O minus there. And so when you put 2,4-dinitrophenol into a cell or in an environment where mitochondrion is found, this 2,4-dinitrophenol will become protonated, and since it is relatively nonpolar, it can cross the inner mitochondrial membrane, effectively transferring protons from the intermembrane space into the matrix, bypassing the ATP synthase. As a result, no ATP is generated in the process, and therefore the electron transport chain, or the operation of the electron transport chain, is uncoupled from that of the ATP synthase. In that case, electron transport is uncoupled from the ATP synthesis. In the case of valinomycin and gramicetine, these are ionophores. They form pores across the intermembrane space. It allows for the penetration of protons, and therefore also uncouple electron transport with ATP synthesis. And this have been used as antibiotics, right? Valinomycin and gramicetine. Notice that they function as antibiotics by actually inhibiting or by lowering the amount of ATP the microbes are able to generate. All right. Okay. Now, there's a quantity called the P to O ratio. P here corresponds to the number of moles of inorganic phosphate, and O, number of moles of oxygen atoms. Right? If you're going to notice the phosphorylation, so when you have the number of moles of inorganic phosphate consumed is actually equal to the number of moles of ATP produced. While in the case of oxidation, rather the reduction of oxygen, you have here, when you say 1 mole of oxygen, when you're talking about 1 mole of oxygen atoms, and you're talking about 1/2 mole of oxygen molecules. Okay, so, and it entails a reduction of O2 by 2 electrons. So these 2 electrons actually come from 1 NADH or 1 FADH2. So when you say PO ratio, number of moles of inorganic phosphate to the number of moles of oxygen, that actually corresponds to the number of moles of ATP that are generated for every 2 moles of electrons that are transferred along the electron transport chain, or for every 1 mole of NADH or 1 mole of FADH2. All right. And we have here a PO value of 2.5 and NADH is oxidized, and a PO value of 1.5 and FADH2 is oxidized. What does that mean? That means that 1 NADH, these values were experimentally determined, by the way. When 1 mole of NADH is oxidized, it produces 2.5 moles of ATP, while when 1 mole of FADH2 is oxidized, it produces 1.5 mole of ATP. All right. Okay. Coupling of electron transfer to oxidative phosphorylation requires a multi-subunit membrane-bound enzyme. It's an ATP synthase. This enzyme has a channel for protons to flow from the intermembrane space into the mitochondrial matrix, and the movement or the translocation of these protons cause the rotation of the epsilon gamma rotator, which then causes a changes in the conformation of alpha beta subunits, which are responsible for the synthesis of ATP. Okay, the proton flow is coupled to ATP production in a process which appears to involve a conformational change of the enzyme, as I've mentioned before, as we're going to discuss in a moment. Okay, now the chemiosmotic coupling, I've been alluding to this already, like I'm sounding like a broken record, right? So in the chemiosmotic coupling, it's based on the proton concentration gradient between the intermembrane space and the matrix. So a proton gradient exists because the various proteins that serve as electron carriers are not symmetrically oriented with respect to the two sides of the inner mitochondrial membrane, and this leads to the transport of hydrogen ions from the matrix to the intermembrane space. These proteins take up protons from the matrix when they are reduced and release them to the intermembrane space when they are reoxidized. Okay, the reactions of NADH, coenzyme Q, and oxygen all require protons. So this is the same flow of electrons, like this is the same diagram that you've seen a while ago, but notice that this time we have indicated the number of protons that are transported from the from the matrix to the intermembrane space. Like the oxidation of NADH by coenzyme Q releases, or rather is accompanied by the transport of 4 hydrogen ions. The oxidation of coenzyme Q by cytochrome C is accompanied by the um, transport of 4 hydrogen ions, and ultimately the oxidation of cytochrome C by oxygen is accompanied by the transport of 2 hydrogen ions. All right? Okay. Now, you're going to ask yourselves, I'm not sure if you've seen that, you're going to ask yourselves, so why then is the PO ratio of the FADH2 just 1.5? It's just 1.5 because you're going to notice FADH2 comes from complex 2, and complex 2 is somewhere here, right? Somewhere here, and it does not involve the it does. First of all, a complex 2 does not involve a transport of protons from the matrix to the intermembrane space. Second is that it bypasses complex 1, right? It bypasses complex 1. So in the end, when one FADH2 is oxidized, you just transport 4 plus 2, 6 protons from the matrix into the intermembrane space. But when an NADH is oxidized, you transport effectively 4 plus 4 plus 2, effectively 10 protons. And that explains why the experimentally obtained PO ratio for FADH2 is just 1.5 moles of ATP are being formed for every 1 FADH2, while there are 2.5 moles of ATP are being formed for every 1 mole of NADH. Alright? Okay. Now, these are the evidence for the chemiosmotic coupling, which was, by the way, suggested by Mitchell in 1961. So they found that system with definite inside and outside compartments, closed vesicle is essential, right? It must be, because you're transporting a proton from the matrix to the intermembrane space, or you're transporting protons from within a membrane to without to that, from the inside to the outside compartment. So there must be some compartmentalization involved in this chemiosmotic coupling. Submitochondrial vesicles can be prepared, which carry out oxidative phosphorylation and have an antisymmetric orientation of respiratory complexes. This just means that you can isolate the portions of the electron transport chain, or some sections of the mitochondrion, and this submitochondrial vesicles are capable of carrying out oxidative phosphorylation. Okay, of course, assuming, and that naturally means that you have the important complexes as well as ATP synthase present in that vesicles. A model system for oxidative phosphorylation can be constructed with proton pumping in the absence of electron transport. How does that happen? Say, for example, when you have bacteriorhodopsin. In this case, the proton transport is not because of the electron transport. The transport of proton from within bacteriorhodopsin to out of bacteriorhodopsin is accomplished by the use of light. Right, light. It is a light-mediated transport of proton from within the compartment to outside of the compartment. Since you generate ATP, since you generate a potential across the membrane, then that also is capable of forming ATP as a result. Okay, a model system for oxidative phosphorylation can be constructed with proton pumping in the absence of electron transport. And the model system consists of reconstituted membrane vesicles, mitochondrial ATP synthase, and a proton pump. The proton pump here is that of bacteriorhodopsins. The existence of a pH gradient has been demonstrated and confirmed experimentally Therefore, the chemiosmotic coupling or chemiosmotic theory is indeed very reasonable. It's already been confirmed. Okay. Now, the mechanism by which the proton gradient leads to the production of ATP depends on ion channels through the inner mitochondrial membrane. As I mentioned before, the protons flow back into the matrix through channels in the F sub 0 unit of the ATP synthase. The flow of protons is accompanied by formation of ATP in the F1 unit of ATP synthase. The details of how phosphorylation takes place as a result of a linkage to the proton gradient are not explicitly specified by this mechanism, but we can of course make some conjectures. So, as you are going to learn more about that, the conformational the synthesis of ATP through conformational changes. For now, what I want you to notice is this. So you have here the matrix, and this is the intermembrane space where you have increased concentration of hydrogen ion. The electron transfer chain, of course, transport the protons here to the intermembrane space. And when this protons, remember, that's natural. The natural movement of substances from region of high concentration to region of low concentration. So when the protons pass through the ATP synthase, specifically through the F sub 0 subunit, then that movement of the translocation of protons lead to the generation of ATP by F1, where ADP and inorganic phosphate are combined to produce ATP. That is called oxidative phosphorylation, of course. That is called the conformational synthesis, the synthesis of ATP for conformational coupling. Okay. The proton gradient leads to changes in conformation in a number of proteins, including ATP synthase. There are 3 sites for substrate and ATP synthase, and 3 possible conformations. You have an open, which has a low affinity for substrate, loose binding L, which is not catalytically active but does bind ATP and inorganic phosphate. The tight binding conformation, which binds ATP. Catalytically active and it binds ATP. These sites interconvert a result of the flux of proton for the ATP synthase. Proton flux converts L to T, which produces ATP. And what is L? Again, not catalytically active. T is catalytically active, tight binding. Proton flux converts T to O, tight binding to O, which releases ATP because O has low affinity for the substrate. So here is what happens. Okay, let's go back to this one. Here is what happens. Again, this is the intermembrane space, and this is the matrix. And remember, this is just a magnified view of this one. Okay, so you have here the protons will enter through from the intermembrane space through this F sub 0 of the synthase, subunit of the synthase, and the flux of the protons will cause conformational change in these proteins. Conformational change in these proteins, which will cause changes in the conformation of epsilon and the gamma subunit. You can think of this epsilon and the gamma subunit like a rotor, like they rotate, right? And such rotation of the rotator will cause the alpha beta subunits to change in conformation. And it is this alpha beta subunits that's actually responsible for catalysis. Like the T, O, and L conformations, that's the one. Okay. B is the one that supports B and A are the ones and delta are the ones that support the alpha beta subunits. So when this rotate, epsilon and gamma change in conformation, or quote unquote rotate, this will cause a change in conformation of the alpha beta subunits, which will transform L to T and T to O, right? And which will then ultimately lead to the changes in ATP. So you see here the chemiosmotic, or rather the potential energy, the proton gradient, is coupled to conformational changes of the ATP synthase, specifically the F1 subunit, which is found exposed in the mitochondrial matrix, ultimately leading to the formation of ATP. How? Specifically via the flux of protons that enter from the intermembrane space into the mitochondrial matrix, right? Okay, as you can see here, so what happens is that the L is converted. Alright, so first and foremost, this L is a loose binding, right? There's nothing in there because it already released its ATP. So it is converted into it. There's nothing in there, right? It's not catalytically active, but it's capable of binding the substrate, so ADP and inorganic phosphate. Remember, the protons are moving from the intermembrane space into the mitochondrial matrix, causing a change in the conformation of a rotator and ultimately the alpha beta subunits, which will change the L, all right, to T. Like this now is a T conformation, and the T conformation is catalytically active. It will catalyze the formation of ATP from ADP and inorganic phosphate, as you can see here. Now, what happened is that the continued movement or translocation or flux of protons will again change the T into an O. All right, this will change into an O, and an O does not have any affinity for the substrate, and it will simply release the proton, rather the ATP. All right, so you have three. You have 1, 2, 3, and these three subunits work in operation to synthesize ATP. All right, I hope that is clear. And each of them are transitioning. For example, this becomes from L to T to O. This also changes from T to O to L. This changes from O to L to T. All right. Okay. In chemiosmotic coupling, the proton gradient is the crux of the matter. The flux of protons through pore in the synthase drives ATP production. In conformational coupling, a change in the shape of the synthase releases bound ATP that has already been formed. All right. Now, how did they study the electron transfer chain? Okay, how did they study the electron transfer chain? They actually used respiratory inhibitors, and these respiratory inhibitors will block electron transfer in one complex, for example, another complex, and ultimately leading to the accumulation of a reduced form of the electron carriers prior to the carrier that has been inhibited, and the accumulation of the oxidized form of the carrier, that oxidized form of the carrier where the inhibition point, or where inhibition has been performed. Okay, respiratory inhibitors can be used to determine the order of reactions in the electron transfer chain. How do you do that? You isolate the intact mitochondria from cells, provide an oxidizable substrate so that electron transfer can occur, add a respiratory inhibitor, and then measure the relative amounts of oxidized and reduced forms of various electron carriers. Remember that there are three sites in the electron transfer chain which inhibitors can have an effect. So what happens effectively, this is the normal one, right? Reduced substrate, oxidized carrier, reduced carrier, and so on and so forth until you reach oxygen being reduced to water. Now, when there's a respiratory inhibitor, for example, so you have carrier 1, 2, 3, and 4, where there is a respiratory inhibitor that inhibits carrier 2, I mean the oxidation of carrier 2 by reduced carrier 3, then you're going to have an accumulation of reduced carrier 2 and an accumulation of oxidized carrier 3, because the reaction between the two have been inhibited by the inhibitor. And so you know that the reaction, or the oxidized carrier 3 must, the reaction catalyzed by the enzyme responsible for the reduction of oxidized carrier 3 must happen after the reduction of oxidized carrier 2. So this time you're able to map the positions of the electron carriers along the electron transfer chain. Okay. As I've mentioned, there are three sites in the electron transfer chain at which inhibitors have an effect, and this is here: the oxidation of FMN by coenzyme Q. That is, remember, part of complex 1. It is inhibited by rotenone and amytal, so it causes the accumulation of oxidized coenzyme Q, because that cannot be reduced. Cytochrome B is also inhibited. The oxidation of cytochrome B by coenzyme Q is also inhibited by antimycin A, and the oxidation of the iron sulfur carried by cytochrome C1 is ultimately oxidized, or rather inhibited by UHDBT. And you have here The oxidation of cytochrome A free by oxygen, it is inhibited by cyanide, azide, and carbon monoxide. Think about carbon monoxide poisoning with people sleeping in their cars, right? Air-conditioned cars. They suffer from carbon monoxide poisoning. Why? Because carbon monoxide effectively is an inhibitor of the electron transfer chain. All right. Okay. Now I hope that you've learned much about the electron transfer chain to oxidative phosphorylation. Now, so far we haven't yet computed for the total number of ATPs that can be produced from glucose, right? In aerobic respiration. All right. So I want to discuss that now, and remember that the actual number of ATPs that are produced in aerobic respiration depends on what kind of cell you have, because it depends on what kind of shuttle mechanisms that the cell uses. And what am I talking about? Remember that all of the NADH and the FADH2 produced in aerobic respiration are generated in the mitochondria, except for one. And what is that? The one which is generated in glycolysis. The NADH that is formed when glyceraldehyde 3-phosphate is oxidized into 1,3-bisphosphoglycerate. It is found in the cytosol, and it has to be transported into the mitochondrial matrix in order for it to be oxidized, right? Okay, so different kinds of cells have different shuttle mechanisms for that NADH, and we're going to discuss them now. The shuttle mechanisms, they transport metabolites between mitochondria and the cytosol. So the glycerol phosphate shuttle, okay, that is the one that is used by muscles. We know that glycolysis in the cytosol produces NADH. NADH does not cross the mitochondrial membrane, but glycerol phosphate and dihydroxyacetone phosphate do. Through the glycerol phosphate shuttle, 1.5 ATP are produced in the mitochondria for each cytosolic NADH. You know why? Okay, let's see. So the NADH from glycolysis reduces a dihydroxyacetone phosphate, which is also produced in glycolysis, into glycerol phosphate. Now that glycerol phosphate is oxidized by an enzyme on the outside of the inner mitochondrial membrane. Once it is oxidized, it produces what? It produces, this is oxidized back into dihydroxyacetone phosphate, which is delivered back into the cytosol, but that produces FADH2, right? The mitochondrial glycerol phosphate dehydrogenase produces FADH2, and remember, FADH2 bypasses complex 1, and so instead of forming 2.5 ATP, just produce 1.5 ATP for every one mole of FADH2, which of course means for every 1 mole of cytosolic NADH, all right? That's the glycerol phosphate shuttle system. Now, that again operates in the muscle. Malate aspartate shuttle has been found in kidney, liver, and heart. So the malate crosses the mitochondrial membrane, while oxaloacetate cannot. So the transfer of electrons from NADH in the cytosol produces NADH in the mitochondria. Right? The transfer of electrons from NADH in the cytosol produces NADH in the mitochondria. All right. How does this operate? So in the malate aspartate shuttle, 2.5 mitochondrial ATP are produced for each cytosolic NADH. How? Okay, so this is what happens. Call it malate aspartate. So here is the NADH that comes from glycolysis that is oxidized by the cytosolic malate dehydrogenase, in the process oxidizing oxaloacetate to L-malate. Malate can cross the inner mitochondrial membrane, so you crosses that. So the malate then is oxidized by mitochondrial malate dehydrogenase of the citric acid cycle, producing NADH. Remember, malate dehydrogenase enzyme, that's one of the enzymes the citric acid cycle, right? So it produces one NADH, which will of course be oxidized in the electron transport chain, and it does not bypass, remember, It does not bypass complex 1. So from complex 1, complex 2, or rather complex 3, and complex 4, you're going to generate 10 hydrogen ions, which amounts or equivalent to 2.5 ATP. Now, what happens to oxaloacetate? Because oxaloacetate cannot be transported across the inner membrane, so it is transaminated by mitochondrial aspartate aminotransferase. It becomes converted into L-aspartate. Do not worry, we're going to discuss more about that when we study the metabolism of nitrogen. So, nitrogen-containing compounds like amino acids as well as the nucleotides. So the L-aspartate is capable of crossing the intermembranes, the inner mitochondrial membrane. So L-aspartate is transported back to the cytosol, which is again, there's a cytosolic aspartate aminotransferase, which will convert L-aspartate to oxaloacetate. All right. Oxaloacetate again ready for another round of reaction or another round of shuttling NADH. Okay, so why is it that certain substances can be transported across the inner membrane, the inner mitochondrial membrane, and others are not? Because these substances have transporter proteins. All right. Okay. All right. So the shuttle mechanisms transfer electrons but not NADH from the cytosol across the mitochondrial membrane. In the malate aspartate shuttle, 2.5 molecules of ATP are produced for each molecule of cytosolic NADH, rather than 1.5 ATP in the glycerol phosphate shuttle, a point that affects the overall yield of ATP in these tissues. Okay? The complete oxidation of glucose, a total of 30 or 32 molecules of ATP are produced for each molecule of glucose depending on the shuttle mechanism. If you're talking about the glycerol phosphate shuttle, then you're going to generate a total of 30. If you're using the malate aspartate shuttle, you're going to generate a total of 32 ATP. Okay, so let us go back again to the synthesis of aerobic respiration. In this case, aerobic respiration, remember, consists of glycolysis, then the action of pyruvate dehydrogenase converting pyruvate to acetyl-CoA, the citric acid cycle, and the oxidative phosphorylation in the mitochondria. So let us consider what happens. So phosphorylation of glucose, this is the ATP reckoning, like how much ATP do we consume, how much ATP do we form. So in glycolysis, phosphorylation of glucose, minus 1, because you're consuming ATP. Phosphorylation of fructose 6-phosphate, minus 1, because you're consuming the second ATP. Dephosphorylation of 2 molecules of 1,3-bisphosphoglycerate, you produce 2 ATP. Why? Because you have 2 molecules of 1,3-bisphosphoglycerate there, right? Because the glucose is cleaved. So plus 2, dephosphorylation of 2 molecules of phosphoenol pyruvate, plus 2. Okay, oxidation of 2 molecules of glyceraldehyde 3-phosphate yields 2 NADH. So you have here 2 NADH here. And the pyruvate conversion to acetyl-CoA mitochondria, 2 NADH produced. So you have here plus 2 NADH. The citric acid cycle, 2 molecules of GTP are formed from 2 molecules of succinyl-CoA, right? Okay, so plus 2 in the and what about oxidation of 2 molecules of isocitrate, alpha-ketoglutarate, and malate yields 6 molecules of NADH, and oxidation of 2 molecules of succinate yields 2 molecules of NADH2, so plus 2. So that case, when you reckon the NADH and the FADH2, remember that you have 2 NADH from glycolysis. This is now the oxidative phosphorylation in the mitochondria. So 2 NADH from glycolysis yields 1.5 ATP each if NADH is oxidized by glycerol phosphate shuttle. So you have here 2 times 1.5. Where did this NADH come from? From the glycolysis, oxidation of glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate. So 2 times 1.5, it's plus 3. While if you use malate aspartate shuttle systems, in other words here, you have 2 times 2.5, it's 5. Remember, oxidative decarboxylation of 2 pyruvate to 2 acetyl-CoA produces 2 NADH, which this is found in the mitochondrion, so that means it's equivalent to 2.5 ATP. So 2 times 2.5, so it's 5, and here is also 5. Remember, 2 FATH2 from each citric acid cycle to produce 1.5 ATP, so 2 times 1.5, 3, and you have 6 NADH. Where did this 6 NADH come from? From citric acid cycle. Remember, you produce 6 NADH because you actually produce 3 for each acetyl-CoA, but you have 2 acetyl-CoA, right? So that's 2 times 3, it's 6, and then 6 times 2.5, it's 15. And when you sum it all up, you get 30 when you use glycerol phosphate shuttle and 32 when you use malate aspartate shuttle. And remember that this glycerol phosphate shuttle operates in the muscle, while the malate aspartate shuttle operates in the in the in the liver. All right, just notice how much more ATP one can generate from aerobic respiration, 30 or 32 compared to the what? To the 2 ATP molecules that can be generated from anaerobic respiration. All right. All right. So there is the end of our discussion on the electron transfer chain. In the later discussion, we're going to discuss the oxidation of lipids as well as the synthesis of lipids. Thank you for listening.